So we had quite a lot of questions coming in, and I think the first one we'll go to probably relates to your talk, Dr. Arlene Drack. So as the retina treatment appears to work with BBS10, will you now be trying the treatment with other BBS types?
So, as with most things in science, we can only work on what we can get funding for. And so I have funding for BBS. Dr. Beales has funding for BBS1. And what we hope is that if we show promise with these types of BBS, that will spur funding to work on the other types of BBS, because these BBS genes all make these proteins that work together. So it makes sense that if we can treat some of them, we should be able to treat all of them. Phil, what do you respond to that?
Yeah, I absolutely agree with you. It's also bandwidth. I mean, it's a full-time job more or less to get these first gene therapies out. And so we would love to work on other ones, but you have to do them sequentially.
Yeah, I agree.
Beautifully shown as well, Arlene, the amount of work that is required before you even consider going into the clinic is absolutely considerable. And a beautiful data that you showed today. Thank you.
Thanks. I look forward to seeing yours. I couldn't quite get up at 2 a.m. to look at your talk.
So this next question, I think, is perhaps for both of you. So someone's asked if there's a difference in the UK and US studies in mice and gene therapy, and they've stated UK no regeneration of retinal function, USA seeing this. If you could explain a bit more about that.
Well, Phil can answer because I didn't see his talk this morning, but that could be a difference in the type of BBS. It might be in BBS10. We can actually turn some of the cells back on, and in BBS1 we can't. It really, like I said at the beginning, we thought after RP65 that it was just all going to work the same way with any retinal degeneration. You just put the gene in and things start working again. But it is really subtle and different between each gene.
Yeah, likewise, we've not seen that type of improvement at all. So there must be a difference between either the actual way in which we've developed the product, the vector itself, or it's a gene difference itself. And it's probably the gene difference, I think.
I think so too. I just want to say that, I was so incredibly thrilled when Phil showed me the data for his BBS1 gene therapy vector, because I had worked on BBS1 before, and it has some important differences from BBS10 because we could not get a good rescue, and Phil has managed to do that. So I definitely think there's a difference between 1 and 10, and I just commend Phil immensely for getting that to work.
Thank you. So moving slightly differently, I think this next question is more for you, Dr. Elizabeth Forsythe. Someone is asking, is there an advantage of setmelanotide in comparison to Wegovy, if I'm saying that right?
I think that's a really good question, right? And I guess the answer is, in reality, we don't know, because we've never done a trial to compare the two next to each other, right? So I guess the compelling bit about setmelanotide is it works on the pathway that we know is not working so well in Bardet-Biedl syndrome, right? So it works on the path, on that specific pathway. And so from a scientific point of view, that's very compelling, because you think actually if you can fix the bit that's not working so well, that's great. And then when we sort of zoom out and look more pragmatically at well, what works overall, we have limited, but we have some data that suggests that actually Wegovy and some of the other drugs that work on a different pathway, primarily the GLP-1 pathway, it looks like they also work in people with BBS. To know the difference, to really know the difference between the two, we'd have to do a big study where we put lots of people on setmelanotide and lots of people on one of the other drugs and then compared the two. I think, I don't know whether that will ever happen. From my point of view, the main the main focus I have is to make sure that we have options for people, because we know from every other drug for every other condition that different people will respond differently, right? So some people might have a really good response to setmelanotide and less so to Wegovy, and the other way around. So from my point of view, the aim is just really to have options for everybody.
Thank you. And someone's asking how you can get prescriptions, or how they can get prescriptions for Wegovy.
Yeah, it's a really tricky one, because one of the things I've realized is that it depends slightly on where you live. The best thing I would say is actually to go back to your BBS clinician, ideally the endocrinologist or the hormone doctor, and they will be able to advise depending on where you're based. And I know, I guess the other thing to say, sorry, is I know that there is a shortage. We know that the people who are on Wegovy, some people have had to have a break, not because they wanted to, but because there was a shortage of supply. I think it will get better over time. I think the industries are having to sort of increase their production to meet the demand of what's available.
Thank you. There's one more on setmelanotide, and that's asking how it is administered in children and what the frequency might be. Yeah.
So it's so I alluded to it a little bit in the talk. So it's what we call a subcutaneous injection. So it's an injection under the skin, and that's the same for everybody. It's a daily injection under the skin.
Okay.
So, but these drugs are all in development, you know, so going from daily injections, maybe hopefully at some point to weekly injections, and maybe at some point there'll be a tablet. Who knows?
Thank you. Someone's asking here about gene therapies, and if you can give an idea of timescales of when gene therapies might be available to BBS1 and BBS10 population. 1 to 2 years, 5 years, 10 years.
I can go first there. I mean, we hope that it's not going to be 10 years, but we don't know how long it's going to be before we can actually start clinical trials. As being explained, there's there's a lot still to get through. You need to confirm safety before you start going anywhere near patients, and in order to do that, you also need to get approval from the regulatory bodies, FDA in the US, MHRA here, EMA in Europe. So in essence, it's there's a lot to be done before you can get to that particular point, because they're the ones who give you the permission to actually go anywhere near a patient. So as I say, I hope it isn't 10 years, I hope it isn't 5 years, but to be exactly sure, we're not entirely certain.
Yeah, I just have to echo exactly what Phil said. And I know he and I are both working every day to try to move it forward, but it is kind of like moving that boulder up the hill. You think you got somewhere and then it rolls back a little, and you have to, kind of do those steps again. So it's in the process, but we just can't say.
Thank you. We've got time for a couple more. So someone states here that there's someone studying neuroscience at uni. They came across a number of researchers looking into prosthetics for RP and other retinal dystrophies, wondering if there was anything promising coming out of research in this area that may help people with sight loss for whom gene therapy might not be helpful.
Yeah, I mean, I'll just say there's so much work being done, lots of really great things. I mean, there are several different kinds of visual prosthesis that can make the nerve fiber layer in the retina sensitive to light. So that's kind of a different kind of vision. The sort of, retinal implants that send a signal, they lay on the retina, and then when light stimulates them, they send a signal through the optic nerve. So all of those things are in development. They've had more and less success in different ways. But I mean, there's nothing that's, I would say, just working really routinely and available to everybody. But there are definitely lots of things in development that way.
Thank you. It's interesting. This is a really interesting question. Are there any countries in the world where there are no people with BBS?
Mhm.
I'm sure that
Anybody know the answer?
I'm sure there are, but by and large, the majority of countries and ethnicities will have BBS, so it's found worldwide.
Mhm.
I'm sure there's a little island somewhere where there's no BBS. Faroe Islands have 1 in 3,000 people have BBS. Very common there. And Sheila's got an answer.
I've got my manual hand up. So I think Phil and I earlier this morning sort of responded to that question as well. But it's probably just worth remembering that given the variability that we recognize with BBS, and we have experience of this obviously for many years in the clinic, there are many patients who go undiagnosed till much later in life. So the ophthalmologists refer patients because they've got the typical retinal changes and a molecularly confirmed diagnosis, but nothing else. So the question often arises, is that actually just BBS with the eye phenotype, or is it BBS because clinically it doesn't fit the spectrum? So I guess there may well be people who have just, who've been well in good health and just haven't had a diagnosis, but yeah, difficult to know.
Thank you.
Once I've got my manual hand still up, I just wanted to say, Dr. Drack, it's really lovely to meet you. And I think you published about RP65 back in 2019, if I remember correctly.
Yes. Yeah.
And the good news is that whilst all the research and the aspects and challenges you've described for trials take much longer than we would want it to, but that is the necessity. It does work out in the end, but certainly in the UK, Luxturna was approved by NICE, and I was very fortunate to be the clinical lead in HST presenting your work. So it's really great to meet you. Thank you.
Wow. That's wonderful. Thank you. And thank you for doing that, because yeah, it's been so great to have that to offer to patients and to see it kind of move around the world. It's wonderful. And each country has had to do their own heavy lifting to get it into their country. So thank you for doing that. And I know that so many people in the UK appreciate it.
That's okay. But it was really, when you're involved in a trial yourself, things in and out. And I certainly personally, I learned a huge amount. And it was just really satisfying that after a process, it was eventually approved for in the NHS and is being used. So thank you.
Yeah, well, that's a great point too, because it can happen, it does happen. And even though in the clinical trials, it's sometimes frustrating for patients because for various reasons, they don't qualify, or, there's age or other restrictions. But then when you finally do get to that approval point, then, it's available for everyone to try. So I think that's the other message as we work through this whole thing with BBS.
Thank you. And I think it's great for our delegates to see everybody working together. As we said at the beginning, we are a global community, and I think the more collaboration there is, it can only improve things for everybody. We have got another question that's come in, slightly different, very interesting. Are there any gene therapy for any other symptoms of BBS, such as urology?
Shall I take that one?
Sure,
I mean, the simple answer is no, not yet. And I think, what's good about this question, it points to urology or nephrology, which is obviously the kidneys. And I know there are lots of groups working on trying to develop cell and gene therapies for kidney problems. It's a huge challenge, because if you think about what does the kidney do, it filters our blood and creates urine. So one of the challenges is if you give a gene therapy there, it's got to stay long enough, and you've got to get it into the right cells that matter. And in our case, certainly in BBS, it's the nephrons themselves, the tubules, that tend to be affected. And of course, I guess one of the issues is going to be, you know, how do you keep it there long enough for it actually to be taken up by the cells themselves? That's part of the problem. But people are working on that, and I think as soon as you know, we see whether or not there are successful ways of doing this, then that whole area, it's a huge unmet need, will start to change.
Yeah, that's a great answer. And I agree. I mean, obviously we would love to just treat the whole body. But you know, as Phil said, there's like a different problem with each organ. Interestingly, I had a graduate student who was working on this. And so I had him do a systemic injection of our gene therapy vector with that GFP protein. So I could see if he gave it intravenously, where did it go? And the only place that really lit up was the eye. So even when we gave it intravenously, it seemed like it mainly went to the eye. Now, some of it might have gone to the brain too. We didn't have a great way of looking at the brain as well, but it just made me realize how hard it is. Because as Phil said, it just goes through. It goes through the liver, it goes through the kidney. It just doesn't, you know, transfect there. But maybe getting better vectors, like if we can get an eye gene therapy trial that shows that you really can correct part of this disease, I think it would make more people interested in, well, let's modify the vector so that it will stay in the kidney cells or the liver cells or, you know, the other places it's needed. I'll just make, oh, sorry, one other comment. I saw that Tim Ogden wrote, and I want to just thank Tim for making this comment and repeat his comment that our BBS natural history study, BBS 10, is not treating anybody. It is just looking at people with BBS at 2 time points doing the same testing, so we can tell which tests might be useful to best show us an improvement after gene therapy. Thanks, Tim.
Thank you. That brings us to the end of the Q&A session. For any of our panelists that can't see the chat or the Q&As, there are lots of complimentary messages in there. Thanking you, all the panelists, for updates and for working on all the various aspects of BBS, doing more and more research and make life better for our BBS community. So got some very lovely messages in there. Thank you.